Yes, madam, go ahead.
Yes, yes, sir. Just a minute. Okay. Good afternoon, all. Dear students, today we are having an eminent speaker, Dr. Mansi, with us. So, ma'am will have a talk on advances in cancer researches around the globe. So, definitely you will get benefit from her talk. So, I request our very own Dean, Dr. Sharik Sayed, to welcome. So, sir, first over to you.
Okay, thank you so much, Professor Jayashree. So it's an honor and privilege for us to be welcoming Dr. Mansi here. Dr. Mansi, Professor Jayashree will give a full introduction, but she's a cancer researcher working on some of the most cutting edge technologies there. And it is a pleasure and honor for us to be hosting her at Anjuman-I-Islam's Technical Campus. So just for your information, Dr. Mansi, Anjuman-I-Islam's Technical Campus is a fairly old trust. It's almost 146-year-old trust. So we have almost 90 plus institutions, 1.5 lakh students across board, a very rich history of doing some very impactful work in the field of education, right from working in with areas of underprivileged students. We have orphanages that we take care of essentially. So, and for us, it's an honor to be working with Anjuman-I-Islam. So, just a little bit of background for you about the institution. So, Technical Campus is established in 2011 with essentially three programs: engineering, architecture, and pharmacy. These are degree programs. So that's how we are based. We are based out of the New Bombay area. So it's really a pleasure and honor for us to be hosting you here. Thank you so much, really, for taking your time out. I know it's early morning for you in Florida, and definitely our goal, essentially, and we continuously strive for that, is to connect our students with leading experts in their field and domain, so that when they are into teaching and learning, they really get a broad perspective about what they learn in their program. And then we try to do that by connecting them with the experts in their field. So this effort is a part of that continuum essentially. So it's really an honor and pleasure for me to welcoming you here essentially, and definitely the students are excited to hear from you. And what we would like here essentially that let's this not just be another just single lecture, but rather an opening for a long collaboration with you on different facets. So I would request Professor Jayashree to take it forward. Post this event, and maybe we can think of some additional workshop or some additional activities around this domain. So with that, without further ado, welcoming Dr. Mansi once again, and over to you, Professor Jayashree.
Okay, okay, sir. So, hi, Dr. Mansi. So, it's a privilege to introduce yourself to our students, and pleased to see you after so long on this platform, professional platform rather.
Same here.
So, I welcome you on behalf of AIKTC family. Dear students, today we have an eminent speaker, Dr. Mansi from California, USA, to deliver a talk on advances in cancer research around the globe. Dr. Mansi is a molecular biologist with a PhD in pharmaceutical sciences with more than 5 years of experience in next generation sequencing, that is NGS techniques, then cell-based assays, imaging microscopy instruments, and analysis softwares, along with the background in oncology, cancer biology, drug discovery, and pharmacogenomics. During her research tenure, she has functioned as an implant trainee with MCure Pharmaceuticals Limited, then teaching assistant, Nova Southeastern University, Florida, USA, then also as a research assistant. So Dr. Mansi has started profession as NGS technical expert in Genesee, New Jersey, USA, and currently she is associated with BioNano Genomics, Florida, USA, as a field application scientist. So welcome Manasi again, and now the session is over to you.
Thank you, Jayashree ma'am. I hope everybody can hear me clearly. I first would like to thank everybody for having me here. And Dr. Syed, I just wanted to say that, the pleasure is entirely mine, and the honor is mine as well to be invited to speak here. So it's completely the other way around. I'm really excited and honored to be invited to speak, and I will be far from being considered an expert because I'm constantly learning new things every day. But I guess it just it's nice to share whatever experience I've gotten until now with the students, and hopefully everybody will benefit from what I have to share today. So I'm gonna try to share my screen with everybody.
Yeah, Sufyan sir,
Yes,
have you
she
given?
can share. She can share.
Yeah, okay, okay, thank you.
Can you confirm if you can see my screen?
Yes, yes, ma'am. Just make a full screen. Yeah, all right, done.
Awesome. So just good evening to everybody who's on the call. Hi to all the students. I will try my best to not put you to sleep because I know it's a long day for you and it's the end of the day. So I'll try to keep my talk short and exciting. So the goal of my presentation here today is not to just give you a core list of new drugs available in the market. I'm sure your Jayashree ma'am has, she's way better at knowing the drugs and everything than I am. What I want to do is leave you with some conceptual understanding as to where the field is going right now. What are the new things that are happening in cancer research and in terms of trying to find out new therapeutic targets for finding new treatments in cancer, and try to leave you with some take-home message as to what avenues you have available to contribute when you graduate from your pharmacy class, get higher education, and actually enter the field of cancer research and give your contribution to it. Because you guys are the next generation of researchers and scientists. So any knowledge I can share with you, any expertise, anything that can help you in that journey, I would be very happy to do so. So moving forward, Jayashree ma'am has given a really good overview of what I do, but I thought because you all are mostly final year BPharm students, as I was told, I just thought I'd give you an idea of my journey as to how I reached here today and my background a little bit and what motivated me to come where I am right now. So as Jayashree ma'am mentioned, my background is in pharmacy, just like you all. I was a Bachelor in Pharmacy student in JSS College of Pharmacy in Ooty. Although it sounds really nice being in Ooty and everything, I assure you it was very rigorous, and I'm really glad to have studied there. After finishing my pharmacy course, I was really interested in research because I used to work with some MPharm students who used to conduct research. I used to shadow them, follow them around, and ask questions. That's where I learned that I'm really interested in research. Pharmacology used to be my favorite subject among everything else that I studied in pharmacy. So I decided to come to the United States to pursue a PhD in basically pharmacology, or they called it drug discovery. But the subdivision that I majored under was molecular medicine and pharmacogenomics. What that is, we will learn in a few minutes, but basically trying to take advantage of genetics, human genetics or even animal genetics, to find new druggable targets and therapies in cancer and in diseases in general. So the entire PhD journey has taught me a lot, and my specialization was in breast cancer, and we'll learn more about that in a minute. But during that journey, I learned that my true passion was it lied in outside of academia and professorship, because I really enjoy talking to people. I enjoy teaching and training people. I enjoy customer support and service as well. So that's what led me to my future role after that as a customer support NGS technical expert at GeneWiz in New Jersey, where I was basically the liaison between our clients and our company. Any service that our company provided in terms of next generation sequencing assays, I was the expert on it, and I used to consult and help customers design their experiments, know what service to use for their particular purpose, and try to help them in achieving their goal in terms of NGS assays. So after that, I had to move to Florida because my husband got a job here. So I decided to move with bionanogenomics as a field application scientist. And I always knew from the time of my PhD that I wanted to be a field application scientist because it's really exciting where you get to travel, you get to visit different customer sites, leading research institutions, talk to leading researchers and key opinion leaders, and actually interact with them, know what they're working on, and be able to train them on the instrument and technology that our company manufactures and markets. So that's what I do on a daily basis. I interact with customers and I train them on our instrument and our technology so that they can use that in their research to answer their research questions. This entire journey has been really exciting for me, but my core journey as a PhD student has always stayed with me, and I really love research. I love teaching. I love talking to people. So the reason I'm here in front of you today is because I have my basic background during my PhD is in cancer research. This is a brief overview of my dissertation, just so you know why you have this strange person that you don't know much about talking to you is I have a research background in breast cancer where I basically studied the progression of cancer from a normal tissue to a tumor on a cellular and genetic level. So I looked at DNA repair and I looked at the tumor and the tissue adjacent to it and compared both of them to see what is actually happening in a normal tissue when it progresses from normal to cancer from a genetic and DNA repair perspective. Now, to investigate that, we used actual patient samples from breast cancer patients, basically early stage breast cancer patients, because they've just been diagnosed and they're not advanced cases where there's so many changes that have happened, which have made it difficult to drill down to the root of the issue or the cause of breast cancer. So early stage was our choice, and I studied mostly stage 1 breast cancer patients, and I used various cutting edge technologies like next generation sequencing, is like specifically RNA sequencing, DNA methylation assays, DNA repair assays, and gene expression microarrays. It will take a whole another lecture to give you an idea of all these technologies, but definitely happy to do so if we get a chance again to talk. So focusing on today's topic, we're trying to find out advancements in research, in cancer research across the globe. I first wanted to wrap your mind around why you're studying the drugs that you're studying. So it's really, it's sometimes very important to bring together the application aspect of what you're studying. So you have a list of drugs, you have a list of class of drugs, but it is very important to tie it together with the biology of cancer and just understand as to why you're studying the drugs that you're made to study. So that is my goal with the next few slides here. So if I had to describe cancer in super layman or simple terms, I would describe cancer as that Bollywood movie villain that you have. But imagine that villain has become such a huge superpower that it can not only evade the police very, very efficiently, it can also multiply itself to create numerous copies of itself, which is indestructible. You cannot kill that villain at all. And it has gotten such superpowers that it will destroy the entire world. Now imagine that level of powers that your movie villain will have, and that's cancer for you. And I'll show you why. Basically, cancer is so destructive and it's really hard to treat because it's a very smart evolved disease. So basically, when a cell becomes cancer, it goes through so many microscopic and genetic changes that it is really hard to reverse those changes. And because of the number of changes that happen together at the same time, it's hard to target all the changes and bring the cell back to normal. It's become so abnormal at that point that it has no choice to go forward and just keep growing. So basically, there was this landmark paper that was published by two researchers, Hanahan and Weinberg. They're very well known in the cancer research field because they have this Hallmarks of Cancer paper that is widely cited by all cancer researchers, because it does a very important thing, which is tell you what a cancer cell does to become cancer from normal. So basically, if you see the flow chart here, you will see some 6 steps that the cancer cell takes to become abnormal. So I'll go through quickly all the steps that we have here, but I'll try not to go too much into detail for the purpose of time. But basically, the first classic feature or characteristic of cancer cells is growing immortally. Like, it just constantly grows without any checkpoints. Normally in your body, every cell will have a specific checkpoint. It'll have a regulator saying, hey, you have grown enough, we don't need any new cells, you have to stop growing now. There are various chemicals and hormones that are released that tell the cells to stop growing. Now, a cancer cell has evolved such that it bypasses those signals, or it just decides it doesn't want to listen to those signals. And they're like, I'm not going to listen to the signals that is telling me to stop growing, and I'm going to keep growing. Now, that evolution is what makes cancer really hard to treat, because it goes out of control of normal bodily functions. Now, that happens from a number of ways and details, which we won't go into right now. But another thing that happens is in a normal body, when cells replicate a certain number of times, they reach a stage called senescence, which means they grow old, basically, just like all human beings do. Cells also grow old and they die. So that's a normal cell for you. But in case of cancer cells, they also say, hey, I'm not going to age and I'm not going to die. I'm just going to keep proliferating. Now, a lot of things happen when cancer cells decide to do that. Cell death, apoptosis, and necrosis are very important regulatory steps or checkpoints that prevent any abnormal changes to get carried forward into future generations of cells, and thereby preventing mutations. So when this doesn't happen, what happens is the cell decides to live on with those abnormal mutations and changes, which get passed on to future generations of cells. And then these accumulate and ultimately push the cell so far forward towards abnormality that it becomes a tumor and shows tumor phenotype more than a normal phenotype. So that's one way. Another way is inducing angiogenesis, which means generation of new blood vessels. So cancer cell needs supply. So if it's going to grow unrestrained and it's going to keep growing continually without any checkpoint, it's going to need resources to grow that. So the way it gets that is by literally making new blood vessels on its own. Usually this process is very tightly controlled and regulated by various checkpoints and mechanisms, but in case of cancer, all those checkpoints are thrown off. So so if you're getting a hang of what I'm trying to tell you, basically anything normal or any laws that are governed for cancer cells bypass all those laws of the body, and they keep doing their own thing. So that's why cancer is very dangerous, and it spreads throughout your body, and it's almost fatal to people who get diagnosed. So those 6 hallmarks had been known for a while, and then recently Henehan and Weinberg included some more causative or hallmarks of cancer that were discovered with more research and emerging studies that scientists carried out. So all these studies, they showed that there are more advanced changes happening as well with the cancer cell, apart from just the basic hallmarks that we just saw. So they are going through these weird energy changes, which means that they are not utilizing energy that the body is gaining in a normal way. So a good example of that is when we breathe, the oxygen in our body helps convert that into energy and glucose. So there's like this whole pathway. In the case of cancer cells, what happens is it shifts to glycolysis, which is basically which basically happens when there is less oxygen and the body does not have enough oxygen. That's like a backup or emergency method of supplying your body with energy when you don't have enough oxygen to sustain yourself. Now, cancer cells make this as their only way of getting energy, which means they shift to a pathway called aerobic glycolysis. So that is again detrimental, and it's usually happens when there's less oxygen, but cancer cells adapt themselves to become that way. So that again leads to an abnormal way of gaining energy and metabolizing and keeping on growing. Of Among all these four, the favorite one, and because I worked on it, is genome instability and mutation. So this was the basis of my dissertation research and my PhD, wherein there are so many changes that are harbored in a cancer cell that it leads to a phenomenon called as genomic instability. So basically, your DNA is a expansive structure of just molecules arranged in a specific pattern, and you all know that probably by now. But what happens is when those specific patterns of bases change, they cause mutations. Now, some of those mutations are viable and they don't really cause harm, even if they happen. But say certain debilitating mutations, they cause mutated proteins, and thereby they cause an altered downstream pathway. Now, one or two other such mutations will make a huge difference in your body, because your body has adaptive mechanisms and DNA repair pathways that will repair that mutation and make sure that the DNA goes back to normal. But now what happens in case of cancer cells is that they lose those DNA repair checkpoints. And this was the basis of my research, is that loss of DNA repair is a very significant a characteristic of early stage cancer. And we found that when we studied breast cancer stage 1 patients, we found that all the tumors stage 1 that we studied in our cohort had low DNA repair, which was really an exciting finding, because what that means is when your cell loses the ability to repair DNA, it basically carries over mutations to next generations. Now, when a lot of these mutations accumulate, that is when your cell becomes so abnormal that it starts showing a tumorigenic phenotype, which is a cancerous phenotype. So it is really interesting as to how all this happens. Now, there are also other hallmarks, but we'll try not to go into too much detail about those. So basically, the whole point of explaining these processes to you is these are the reasons why you have these drugs in front of you. So if you're seeing each hallmark of cancer has been used as a target for a specific therapy for treating a specific type of cancer. So it's not necessary that all cancers have all these features, but there's always a combination of these features going on, and there's a driver change that basically drives the transformation of normal to tumor, and there are passenger changes, which means that there are changes that have to occur along with driver changes that just carry forward with the future generations of the cells. So any of these changes can be the major driver of transformation, and then there are other changes that happen either because of the driver or independent of the driver that lead to overall an abnormal phenotype and an abnormal behavior of the cells and then the tissue. So going into each of these classes of drugs is again an own lecture of itself. So we'll try not to do that for the interest of time, but some really interesting discoveries have been made in the recent times just by taking advantage of these specific characteristics of cancer cells and the hallmarks that they show. So coming to some really interesting cancer therapies that have been developed in recent ages, we'll try to focus on just a few of these just in the interest of time. There's a lot of new things going on, and everything is so exciting, but I'll give you some brief snippets based on what I have worked with and what I have experience in. So Let's first focus on, just to give you a brief idea, and just to make sure everybody's on the same page. You all are aware of the central dogma of molecular biology, which is basically the whole process of DNA being converted to RNA, RNA being converted to protein, and protein being the ultimate effector molecules that go and carry out the specific action that is required from that protein. So that is the whole, the central principle of functioning of the body. So your DNA is basically your code. It basically tells you, or it's a set of instructions, right, for your body. Instructions as to what to do, what RNA to make, which will make a specific protein, how much of it to make so that it will make just enough and not too much or too little, which will affect normal function, which is very, very important in cancer, in diseases, in just the normal functioning. The quantity, the actual protein that is made, and just the normal functioning of the DNA and the processes therein is pivotal to making sure that there is nothing abnormal going on. So now, as you know, the conversion of DNA to RNA is called transcription, and conversion of RNA to protein is called translation. Now, there's a really interesting way that was found where RNA can be converted back to DNA by just reading the bases and just finding complementary bases to those and trying to go a step back. And that is basically called reverse transcription, and is done by certain enzymes called reverse transcriptases. So those enzymes are really important in certain very, very frequently used technologies like polymerase chain reactions, or PCR. PCR, by the way, if you're not aware of it, is the basis of the tests that have been made to diagnose COVID-19. So right now, all the COVID-19 tests that are out there that detect whether you are positive for COVID or negative are all PCR based, which basically use these reverse transcription enzymes to detect the presence of viral RNA in your blood and in your cells. So just thought that was something really exciting. So the reason I explained the central dogma to you is it's very important to understand what the normal process is, to know what this really small pocket-sized powerhouse does in your body. So we're going to talk about microRNAs, which are also called miRNAs. MicroRNAs are nothing but very small single-stranded fragments of RNA. They're just about 20 to 22 nucleotides long, so they're super small when you consider the entire, say, an RNA transcript, or when you consider the length of the DNA. These small nucleotide molecules, they are basically inhibitory in nature. So what they do is they work by either two ways. So they either inhibit the process of translation of RNA to protein by literally binding to the translatory unit on the RNA and thereby preventing translation, or they literally go and chew up the RNA. So they will degrade that RNA so that it cannot be translated into protein. So miRNA has been, like, it normally in the body, it functions as a regulatory mechanism, which means it will regulate as to how much of the protein to produce and when to produce and all that. So it's basically a checkpoint in your body. So saying, okay, we now have enough amount of this protein, stop producing it. That's when the signals will be generated for release of that microRNA, and then blocking of the RNA to stop producing the protein. Now, these microRNAs are sometimes overexpressed or underexpressed in cancer, and they exhibit either oncogenic properties, which means they become cancerous, which means they promote cancer, or they exhibit tumor suppressor properties, which means they are inhibitory in nature. They inhibit the production of certain proteins that will promote cell proliferation and all those abnormal changes that happen in cancer. So recent research is taking advantage of this naturally available molecule in the body to make it into a therapy. In which sense, microRNA is used in therapy can act in either two ways, right? If you have a microRNA that usually inhibits, but we needed to, we needed to keep working, we just inject more of the microRNA so that if you need it to inhibit an oncogene, the microRNA injected will go and bind with the oncogene and will suppress the oncogenic or tumor promoting activity that's going on. If a microRNA is oncogenic in nature, which means presence of the microRNA actually activates the oncogene, there's another approach that's used, which is literally having an antisense or an anti-miRNA produced, which is a small RNA molecule, which is exactly complementary to the miRNA. It's also called an siRNA. So what that does is it literally binds to the miRNA and stops it from binding to the RNA, and thereby stops it from inhibiting the RNA. So there's multiple ways that researchers are taking advantage of the miRNA activity. And I actually had the fortune to work on an miRNA study during my PhD, because a colleague of mine, his PhD dissertation was on microRNA. So super interesting topic, and a lot of people in cancer research are, especially in biological sciences, are working on microRNA assisted therapy. Okay, another beast in the cancer research field is the CRISPR-Cas9 gene editing system. Now, I'm pretty sure if you follow any kind of medical research or cancer research, you have definitely heard of CRISPR-Cas9. And I myself have never worked on it, but it's so hot that I had to definitely talk about it. It is really one of the most cutting edge technologies that every cancer researcher is using in their research, and is trying to use the power of gene editing to either create knockouts, which means just removing the gene of interest and then seeing what the effect is after you remove that gene, or just creating, just editing the gene, wherein you either add a fragment, or you just edit a bit of the gene to remove the active fragment or a specific exon of the gene. By and making these manipulations are helping in brainstorming possible therapies in or gene-based therapies in cancer. So for example, say you have a disease where you have say muscular dystrophy, where there's a specific repeat region on DNA that is contracted, right? Basically it should be longer, but because it shortens, it leads to certain manifestations of deformities and muscle weakness and so on and so forth, all the symptoms that you see in muscular dystrophy. So what you can do basically in CRISPR-Cas9 is basically identify that region, add in those repeat regions, and then make sure that function goes back to normal. Now, it sounds really easy, right? Just take some cells, add the gene, and you'll be looking at a completely cured patient. No, it doesn't really work that way. The reason that CRISPR-Cas9 has been really controversial and has not been hugely successful as much as how much it was anticipated is because although CRISPR-Cas9 is revolutionary in terms of just its capability and the applications that you can put it to, it is also it also comes with a lot of limitations. So for example, the basic target identifier sequence is just about 20 base pair long. The spacer that you see here that's the one that identifies your target, the target on the DNA that you want to modify. So imagine that 20 base pair sequence needs to be completely unique, which means there has to be no other region on the DNA that shares even remotely any sequence of any nucleotides of that sequence of the spacer. What happens if there's a shared sequence is that you will see something called as an off-target effect, which means you now have the spacer binding to two regions in DNA instead of just one region that we were interested in binding it to. So what happens is it edits both these regions, and there are some unknown effects of editing that second region, which there's no way of predicting, because there's hardly any precise prediction method that people have developed which can predict as to what will happen when this fragment goes and binds somewhere off target, because you will essentially be able to predict in a very limited fashion as to what the off-target effect might be. So doing this in a petri dish in a lab is fine because it can be tested. But when you give these to patients and when you have unknown effects of a treatment method, it can lead to a lot of complications, side effects, and also really deleterious changes that you don't want to see in your patients. So it's even though it is revolutionary, it has its own limitations. And to date, in the US, only 3 patients have been known to have been successfully treated with the CRISPR-Cas9 editing mechanism. They were, I think, sarcoma and leukemia patients, if I remember rightly. These were the patients that did not respond to any other treatment, and that's why they decided to enroll them in this experimental technique. They saw really good promising results wherein they saw these patients were disease free for about 9 months. They were able to survive for 9 more months, where they had no hope of surviving with the traditional method. So it definitely has a future, but it needs a lot of work and research to make it safe for patient use, and also to make sure we can accurately predict and eliminate the off-target effects of this technology. So let's see. This is my favorite topic, precision medicine, because this also formed the basis of my research, and this is the application of my research on what I worked on. So precision medicine or personalized medicine is also taking a strong hold in cancer research right now, because imagine giving imagine you were told that, okay, you like ice cream? Okay, there's just vanilla. There's no other flavor because, we just decided not to make any flavor. You won't be very happy with that store selling that ice cream because say you like chocolate or you like strawberry or something like that, and there's no, you don't have the ability to make a choice. Now, ice cream is a very trivial matter, or it's not that important, but when it comes to treatment, one treatment cannot be applied to every single person because of a lot of reasons. Primary reason being difference in the type of disease, difference in the say molecular or genetic characteristic of that disease, difference in the individual themselves genetically. So as you know, all of us are genetically very different, even though we are majorly genetically similar, which makes us all humans and different than other species. But still, what makes us different individuals is our genetics, and that plays a huge role in how these patients respond to treatment, how what treatment is effective, what treatment is not effective. So precision medicine has really opened up a really nice arena to treat patients with treat patients based on a subcategory. So the best example that I can think about is, say, breast cancer patients were treated with their usual. They used to do, depending on the stage, they'll do a surgery, they'll do a tumor removal, they'll give you radiation on the surrounding area to make sure they have gotten out all the cancerous cells. And they will just give you a basic empirical, that's what they call it, empirical chemotherapy, which is standard for all breast cancer patients. Now, doing this did not take advantage of specific classes and types of cancers that you usually see in breast cancer, which was discovered at a later time. And this discovery showed that there are certain molecular characteristics of breast cancer which can be which will help us classify breast cancer into different types. So there were estrogen receptor positive breast cancer, there were estrogen and progesterone receptor positive. So they're either hormone receptor positive or hormone receptor negative breast cancer. So researchers realized that the breast cancers that show a specific hormone receptor on their cells, they can be manipulated to basically develop an inhibitor for that hormone receptor, which will help us treat that specific type of cancer better. So a really good example is treatment of HER2 receptor positive cancers with trastuzumab or Herceptin, which is the brand name here, or even treating hormone receptor positive cancers like estrogen receptor positive with tamoxifen, which is a very well-known and commonly used drug for breast cancer patients that show hormone receptor positivity. Now, there are also a category of patients that are hormone receptor negative, which means their breast cancer type is such that they do not show any overexpression of hormone receptors on their cells. Now, as you can imagine, there's nothing to target, or it's really hard to target anything on these cells. And that's what makes treatment really hard and complicated for these patients. So hormone receptor negative or triple negative breast cancer, if you've ever heard that term, is very, very hard to treat and is usually very aggressive. So you see how dividing these into subtypes and treating them based on their characteristic is a really powerful technique. And now people are going even one step further, and instead of just using precision medicine, they're using personalized medicine, which means basically just say using next generation sequencing to sequence a patient's entire genome and seeing if they have like a particular mutation or a particular sequence that will make them either too sensitive or less sensitive to a particular drug. And adjusting the dosage based on that. So that's like a very advanced application of personalized medicine, which will definitely be in the clinics very soon, because that is the level of differences that you usually see in human individuals. And that difference makes it really hard to treat patients with the empirical therapy. So introducing personalized medicine is a really exciting arena for a lot of clinicians. However, it takes a lot of work to do personalized medicine. It takes a lot of informed geneticists to be employed in every hospital to be able to explain what those genetic results mean to the doctor as well as to the patient. So it's an added expense. It's definitely not very easy to have these genetic tests done and then just prescribe a medicine based on that. You have to understand what all those results mean. The bioinformatics aspect of sequencing is really complicated. Not everybody is capable of understanding it. So it kind of limits the application of these genetic testing techniques in the clinic. It also limits the, it also requires certain equipment, which is A, very expensive, B, also complicated to operate. You need really specialized people who have experience in it and know what they're doing when they operate these sequencing-based genetic testing methods. So all in all, it is definitely possible and it is looking promising, but it's going to take a lot of work and a lot of more research to bring this science from from the bench in a lab to the clinic, to the bedside of the patient. So this is my last slide today. I'm going to end with just giving you, just putting some conceptual understanding in your mind as to why cancer is so complicated. And this, when I show this to in my talks, I tell them this is the most important slide in my presentation. If you've understood this, you've understood cancer. And if you understand cancer, you will never take it lightly, and you will never say, oh, there's a cure, because it's very hard to find a cure for cancer. And this is the reason why. So basically, cancer was known to be a multi-step process, which means there's a stepwise addition of changes, abnormal changes that happen to cells. And then these abnormal changes keep multiplying when the cells divide. And then you have these fields of abnormal changes that are at varying levels. And I'll tell you why in a minute. And then you have these field carcinogenesis going on, where one group of cells is more forward in the process than the other. And then there's a final group of cells that gets the final change that drives its phenotype or the way the cell operates from normal to tumor. And that is what is the tumor cell here. So imagine this is your normal tissue right here in pink in the top line. This one green cell gets one mutation. That is the first mutation in the series of mutations that would lead it to cancer. So that's your green cell. Now this green cell is going to divide. It's going to form a population of its own. And because this green cell already has a mutation, it is more susceptible to getting further mutations. So among the green cell, there's this orange cell that gets the second mutation. Now you have two populations, one of the green cell that has the first, which is already dividing, then the second orange cells, which is now again dividing, forming a second population. Now, this process continues where there's a yellow cell among the orange that gets the third mutation, the fourth mutation, and so on. So there are these fields of different cell populations formed with varying degrees of mutations that will ultimately accumulate in these blue cells at the bottom, and they will transform from normal to tumor. Now, imagine having all these precancerous cells in your tissue, in your organ. And this is very trivial because this is just a few cells you see here, but in reality, there are millions of cells. The rate at which mutation happens is 1 in a million. That's the average rate of formation of a mutation. So you have a million cells with that one mutation until you get the second one. You have another million until you get the third one. So imagine those millions and millions of abnormal cells sitting right there along with your tumor. So that's why it is so complicated. Now, the other cells, apart from the tumor, when you take the tumor out, you've basically taken the abnormal looking cells. However, genetically, there are a lot of abnormal cells even around the tumor that was taken out, which, if they get the future mutations, will ultimately, they have a high risk of becoming that tumor. So cancer is an extremely complicated topic, and it is very important to understand the complication, and that is why we have multiple modes of treatment. We have multiple therapies that are used together. This is all in the hopes that we will be able to target all these abnormal cells that don't have the normal function, get rid of them, and help the patient get into remission and help their survival and increase their survival after the cancer diagnosis. So I really hope that I tried to keep this short and sweet. I hope that I was able to give you some new information and really instill into you how important it is to keep working on cancer research and keep finding new things, because everything you find is a new knowledge for someone who probably at some point will know how to very effectively treat cancer and put patients into remission for a long time. So I know if we can't get all your questions answered today, I'm always available on LinkedIn. You can find me on LinkedIn. Make sure you drop in a note to me so I know that you heard me talk here today and that's why you're reaching out. Do connect with me on LinkedIn. Any questions you might have on, say, a career choice or cancer research or any questions on today's talk, I'd be happy to answer. I will also leave my email with Jayashree ma'am today. And if you ever have pressing questions or if you need advice, anything that I can do to help you, I would be happy to do that at any point of time. So I'm really thankful to Dr. Syed, Dr. Sheikh, and Jayashree ma'am for having me today. It was really exciting to talk to you guys and just share what I have to share from my experience. I hope you took something useful out of this, and thank you so much for having me. And if you have any questions, how much ever time we have left, I can help you answer those questions.
Yes, thank you, Dr. Mansi. It was really a very nice session. Ma'am, one question from our student is that, what are the major reasons for converting normal to tumor cell?
Sure. So yeah, that's a really good question, and that's the question researchers have been still trying to answer. It's really hard to pinpoint that one tipping point where that cell got converted. So it's basically a very gradual change where you, as I said, there's two kinds of changes or mutations that are known to happen. There are driver changes and there are passenger changes. So driver mutations are the ones that are known to tip the point from normal to tumor. So examples of these driver changes are mutations in very important oncogenes or tumor suppressor genes. If you've ever heard of P53, P53 is a tumor suppressor gene that has very important functions in our body. And most of cancers, you will see a mutation in the P53 gene, which means that the function of P53 is affected in some way so that it loses its tumor suppressing ability and kind of lets cancer develop and evolve. Similarly, there are oncogenes like RAS and MYC. These genes are basically their function is to promote cell growth, proliferation in a normal setting, but there are also checkpoints. But then if the promotion is too much, like if the stimulation to keep growing is too much, which is in the case of oncogenes getting mutations, that's when there's that tipping point from normal to tumor. So again, very hard to pinpoint, but researchers know some major driver changes that have led to the transformation.
Okay, thank you so much. Ma'am, next one is cancer therapies usually come with many side effects like hair loss. So what researchers are going to reduce them?
So that's a great question. So let's try to understand why you see hair loss. So basically, and this is really exciting because this opens up understanding as to how cancer drugs work. So the reason you see hair loss and stuff with cancer drugs is that most cancer drugs usually attack some form of replication, which means they attack the most important characteristic of cancer cells, which is to keep growing. So to keep growing, they have to keep replicating. Now, if you attack their ability to replicate, that will be a major hit and you will stop the cancer to progress. So when you do that, you're not just giving treatment to the cancer cells. You're not just focusing or injecting that medicine to the tumor, right? You're treating your whole body because you are taking a tablet or a drug that goes through your bloodstream. So what that does is when it affects your cancer cells, which are rapidly dividing, it also affects your normal cells in the body that are rapidly dividing. One of them is your hair cells. So your hair grows because your hair cells are rapidly dividing and growing. When you give cancer treatment, it also affects that rapid division of your hair cells. So this is something that is a side effect that can be dealt with, rather than, it's not a debilitating side effect that will affect the health of the patient. So it's something that is not a priority, I would say, to treat that, because on the other hand, you are seeing cancer cells being hit and killed, and the patient living longer. So hair loss is a very temporary thing, which when the treatment is stopped, it will come back. So the treatment is stopped when they have enough confidence that they've given enough dose for the cancer to go away. They do screening, and then they find out, okay, there are no more tumors, or there's the tumor load has decreased, and now we can stop the treatment and you'll have your hair back. So they're discovering new therapies that are not targeting replication, but then they're targeting other mechanisms, as I mentioned, like say angiogenesis. So they're targeting formation of blood vessels. They're targeting formation of certain hormone receptors. So when multiple mechanisms are combined together, that's when you have an effective therapy. So if a patient is seeing really harsh side effects with, say, an alkylating agent, the doctor will probably not give that and add other two modalities of treatment to the patient, which the patient responds better to. So this, again, we're delving into an area of personalized medicine. So it's coming, but it's definitely slow.
Thank you so much.
Can I have asked a question, Jayashree ma'am? Also, I'm
of
also
course,
a student
sir.
out
Of course, sir.
Mansi, a quick question.
Sure.
If you look at the lot of these new therapies, and I've worked on even oncology.
Yeah.
Number one is I'm not talking about something like PD-1 or others, but most of the times you see a small increase in progression-free survival or marginal 6 months improvement over standard therapies.
Yeah.
And on the flip side, you see these huge cost of these drugs. Most of them are into monoclonal space. And so if you translate that for something like a country like India, where, most of it is in a out-of-pocket kind of thing, there's not much of an insurance coverage. What kind of benefit that you look for essentially, and is it helping countries like ours essentially? When you look at the cost and the relative benefit that gives, essentially, when you're talking of just additional couple of months of survival. So what's your philosophical thoughts on that?
Yeah, that's a really great question, and it's a very loaded question, Dr. Said. So I think it's a fine balance, and I think and correct me if I'm wrong, the question you're asking is it worth it to, go through so much for just such a short survival time if that's all you're going to have? And
I mean,
the reason, go ahead.
yeah, and that, and plus the whole idea of FDA approving that and not looking at the cost perspective of that.
Yeah.
I think that's also something that bothers me. That we
Yeah.
put a lot of burden on taxpayers and payers for a very small thing that, and that still incentivizes the companies to continue doing what they are rather than going for major breakthroughs, something like what
Yeah.
you see for hepatitis and others in the last couple of years. So just some thoughts on that.
No, I absolutely see where you're coming from. And the answer to that, I think, is there's a common thinking that there is no cost to life, right? So when it comes to saving your loved one, say if you have a family member struggling from cancer, you're ready to put whatever it takes or whatever amount of money it takes to get them that treatment and get them at least those few moments of life with them, right? So it's really hard. It's from an ethics perspective, it's hard not to be able to justify just helping patients survive whatever short duration that the drugs can buy them. So the ultimate goal, I think, that the field is moving towards is to keep on developing therapies so that time of survival keeps increasing. So to reach that goal, I think these are the incremental steps that are being taken. So right now, for example, with CRISPR, they see, by giving those treatments, they see about 9 months of survival. Now, you may say that's not much considering the lifespan of a human, but for a patient who had no time for survival, like they were possibly given a death sentence, to a patient who got almost a year of survival, that from a patient perspective, that's like a huge change. So I think that is what drives these research and the studies and the FDA approvals that you're talking about, even though it takes billions of dollars. So it's just, you can't put a price tag to life. I think that's the way I see it. So but I totally agree with you. Like it's a very ethical question and a philosophical one too. That would you rather suffer 9 months, or would you? It's a very difficult topic, but a loaded question. But I really would love to engage with you on that, maybe offline.
Yeah, maybe sometime.
Yeah, absolutely.
Thanks. Okay, go ahead, Professor Ashri. I'm done as a student.
Okay. Any questions from the students?
One thing also I think might help students to understand, they especially with a lot of GI, we've talked about tumors and others. There's something also in GI tumors where you want to expand that to students, that especially with old age, and a lot of this happens a lot in India. There's recurrent infections, and that infections can sometimes take a shape of a cancerous state where the cells are trying to repair themselves, and as this continue to divide and repair, they get these abnormalities inside. Can you expand that? I mean, that might help students to connect what they see typically around them.
Sure. So a great example of that, and I'm glad you brought it up, is cervical cancer. So cervical cancer is basically caused by a virus infection. So HPV virus, or human papilloma virus, that's what it's called. They now have a vaccine for it. So they recommend that usually any female about, I think it starts from a very early age. I don't remember the exact age that you can start getting the vaccine. I think it's from age 16 or something, but you can really protect yourself from a very early age and make sure you don't get infected with HPV, because it again comes down to making sure you have safe practices and other things. So it definitely ties to making sure you follow safe practices and make sure you're up to date with your vaccines. They're there for a reason. So if I have anti-vaxxers in this community, in this talk right now, that's going to be a major problem for me. But very important to make sure that you understand why you are being told to do a specific thing. So making sure you prevent yourself from HPV is making sure you prevent yourself from cervical cancer. Now, that's not saying that you would never get cervical cancer if you never get HPV. There are other causes of cervical cancer too, but when you reduce your risk of catching an HPV infection, you reduce your risk of catching a risk of getting cervical cancer. So risk is a difficult concept for people to understand because it's not a cure. It's reduction in the probability of you contracting that specific outcome. So make sure you reduce the risk. And yes, definitely, infections are a way that cancer can develop, and not every infection can lead to cancer, but there are certain examples, especially the one that I just gave. It is prudent to make sure you stay up to date with how to take care of yourself and the best practices that you should follow in terms of your health.
That was nice. So with this whole pandemic coming up and everything is talking about coronavirus vaccine research, would you inspire my students to take up something very impactful? Any dose of inspiration for them?
Dose of inspiration. The dose of inspiration for them, I think, it's just that biology is so complicated. I mean, the reason that they have to just having a conceptual understanding of how things work and why things the way they are. So the biggest advice that I would have for students or as inspiration is ask questions. Ask why. It's like, why does it take so long for a vaccine? Why does it, why is coronavirus so hard to treat? Why is it so easy to contract the infection? So when you ask questions, when you ask why, you know the reason, and there's no better inspiration than knowing the reason behind something. So for example, the reason that for right now, if we talk about the US is facing shortages on testing. So apart from the political side of it, where people are in some places trying to suppress testing because apparently it needs to increase in the number of positive cases, which is intuitive, but whatever. The reason that it's hard to make sure testing is widely available because, as I said, it's a PCR based test. So PCR, if you just take one PCR reaction in a 96 well plate, I used to do PCRs for our miRNA studies. So the one PCR will take you about 2 hours. Now imagine you can't just test patient sample and you're done. You have to make sure you have the appropriate controls. You have to make sure you have a negative control and a positive control to make sure you're getting the right result. So this is patient results you're talking about. It's not just a lab test. So if you're testing patients being positive or negative, your test will determine what happens to that patient, whether they get quarantined for 10 days, separated from their family, getting treatment and on a ventilator, or they go home because their test is negative. Now, if you send home a patient that's positive but came back as a false negative, you are putting a lot of people in jeopardy. So the importance of that test is it cannot be trivial. So another thing is the reason that they're also trying to find out faster methods of testing, because apparently PCR, even though it's a 2-hour reaction, generating that PCR test, generating the report, trying to communicate it to the doctor, that's a long process. So it takes multiple PCR reactions to process just a handful of patients, and that takes time. And there's so much backlog, and that's why there is a backlog on testing. So now there's a talk of talking about, say, antibody-based testing, which is basically just, say, like a pregnancy test you would take, would just test for a specific presence of a specific protein or an antibody in your blood. So all you would need to do is just take a drop of your blood, take the antibody test it, and you would know if you have the antibody for a virus, which means you already were infected and now have the antibodies. And if you don't, which means, you haven't been infected yet. That is something that is now being developed for a quick test. But now there's pros and cons to everything. A PCR-based test is more sensitive, but more time-consuming. An antibody test is very quick, but it's not that sensitive. So there's a high probability of false negatives and false positives. So there's so much to consider. And the fact that we have tests available in spite of these considerations just tells you how far research has come and how important research is. So because researchers are working round the clock, you have these tests ready to be administered, ready to be given to you if you ever have the misfortune of getting that virus, and ready to, you know, be treated if you have symptoms as well. So imagine you being in the forefront of that pandemic and research and trying to find out answers as to how to do this properly. And it's a great thing to do, and you should definitely try to strive towards being, being a part and doing your part and contributing to society.
Great, thank you so much. It was very nice.
Yeah.
Yes, Professor.
Yeah, Mansi, there are a few questions.
Sure.
Can cyst be converted into tumor?
That's a great question. So that's a distinction between a benign tumor and a malignant tumor. So what happens is the difference here is the properties of the tumor cell. So can it be converted? The answer is yes. Will it be? We will never know. And the reason being that think about like a cyst is basically nothing but just uncontrolled cell division, right? You have a mass of cells that have just grown and are sitting there. Now, a cancer really becomes a problem when it gains its characteristics of invasion and metastases. I think I probably skipped over that in the interest of time, but metastases is a very important concept in cancer, and that is what makes it a really big enemy and hard to treat, is because you can easily keep removing if it's growing in one place, and if it's proliferating, going in a mass in the same place, you remove it. When it grows back again, you remove it again. So it's a fairly logical and easy concept. But the characteristic of metastasis is when cancer cells gain the mobility to leave that place or that location and travel outside of the tissue into your bloodstream, into other organs, and across throughout your body. So when it does that, that's when the cancer becomes a problem. So it becomes very hard to treat, because now you have invasion of cancer cells across your body, and you have to basically blast your body with a lot of chemotherapeutic drugs, radiation, etc., to be able to make sure you have gotten rid of cancer cells throughout the body, which you can never be sure of. I mean, there's definitely no way that you can ever be sure that you've gotten rid of cancer. So a short answer to that question is, can a cyst become a tumor? Yes, it can. Will it become a tumor? We don't really know, because if you were able to remove that mass of tumor in time, and if there are no leftover abnormal cells, which may become a cyst again and a tumor and then metastasize into your organs, then surely it can be treatable. So that's why having a benign tumor or a cyst is better, because it's treatable, it's not malignant, and it hasn't spread to your body.
Yeah, Manasi, and the last question: why cancer cells doesn't show symptoms in early mutation stages?
That's a great question. So what happens is, when your cell develops a mutation, your body is already smart. Your body knows that there's something wrong. Your body has already developed mechanisms so that it can correct that mutation, make sure that it's reverted back to its normal state, and then allowed to replicate. So DNA repair here plays a huge part. So basically, your body is telling the replication cycle, hey, stop here, because I see there's a problem. I need to repair that problem, and then you can continue with your cell division. So that's literally what happens, but it happens in the time frame of milliseconds in your body. So in milliseconds, your body stops replication, repairs that damage, carries on with replication. So that's a normal body function. But what happens in the case of cancer cells is that mutation gets acquired. Replication is not able to identify that there is a mutation, right? Or maybe there is no repair process. The repair process has been impaired, like it happens in preliminary stages of cancer. Now, these are such subtle changes in your body that they don't manifest as symptoms, because for you to get a fever, you have to have a full-blown infection and immune response in your body for your body to indicate to you that you have an infection and you therefore have a fever. So behind the fever, you were infected already 10 days ago. Let's take the example of COVID-19. People can stay asymptomatic for a while before they start to show symptoms, or probably will never show symptoms. So symptoms are your body's way to tell you that there is something really wrong. Treat it right now. Whatever happens before that is so subtle that your body is not able to tell you, or it doesn't show up morphologically, or either, there's no external sign that there's something wrong. Now, if you already, say you were a psychic and you knew that you were going to get breast cancer, and you removed a tissue from your breast and tested it under a microscope, a pathologist will be able to tell you that these cells are abnormal, because certain abnormal cells undergo some certain phenotypic or morphological changes, like their nucleus becomes really big. Their nucleoli, they multiply, and there are multiple nucleoli in their nucleus. Their cell to nucleus ratio changes. So those are the things that a pathologist will look for, and they'll be able to tell you that they're abnormal changes. But by the time you know to check for breast cancer, it's probably too late, because you now have a tumor, a significant size tumor. So that's only when, so it's just the problem of scale and sensitivity. Like, you cannot predict that you're going to get cancer, so you never know to check for it. There may be symptoms already, but it's really hard to know that you have it until you see an external symptom. And that's why, one more thing that I wanted to stress here is that's why preventative care is very important. For example, in breast cancer, every woman above the age of 25 should get a mammogram every year. And that is because a lot of times, last year, women have seen nothing, and the next year in their mammogram, they have seen lumps. Now, that 1 year was enough for their breast to develop that tumor and actually show up in their mammogram. Imagine someone skipping that mammogram and then trying to waking up one day to find out they have advanced stage breast cancer because they skipped their mammogram. So it is very important that you do your wellness checks, your health checks, all those that your doctor recommends. Do a regular pap smear, so you make sure you are cervical cancer negative. There's a lot of things that you can do preventatively to make sure that you don't miss the signs and the signals that your body is trying to give you.
Okay, thank you so much, Mansi. So actually, due to time constraint, we will stop here. And there are few questions, but definitely I'll forward it to you.
Yeah.
Yeah. Okay.
That sounds good.
Yes. Yes. So, so it's just a last portion for this webinar. Just a vote of thanks. So I, Professor Jayshree Patil, on behalf of AIKTC School of Pharmacy, New Panvel, extend a really hearty vote of thanks to our expert, Dr. Mansi, who spared time from her busy schedule to deliver a talk on a cancer research for our students. Today, we had an opportunity to hear your thoughts, research aspects in the cancer field, specifically sequencing platforms, and this will surely be going to encourage and upgrade the knowledge of our students in future. I extend my thanks to Dean, School of Pharmacy, Director, Kalsikar Campus, and the management for their consistent support and motivation. My personal and sincere thanks to Professor Abu Sufyan. And finally, I would like to thank our budding pharmacist. So thank you so much for being with us, Dr. Mansi, and looking forward to see you again.